Welcome to this Information Day on the Accelerator Challenge, novel biomarker-based assays to guide personalized cancer treatment. I'm here with my colleagues Jordanos Arsimanoglu, who is the Program Manager on Health and Biotech, and the Policy Officer Vilma Radvilaite, who is a Project Officer in the Accelerator Unit. And they will present to you today all information that you need to know about this challenge. We will make it available after the end of the event on the same event page to which you connected now. And the slides will also be put there. Today is your opportunity to ask all questions to Jordanis and Vilma about this challenge, and you can do that through the Slido. So join at sly.do and put the event code challenges. And there you can ask your questions. And after the initial explanation by Jordanis, he and Vilma will answer all your questions that you have. So I will start today with a short introduction about what the European Innovation Council is and how it distinguishes itself from other programs in Horizon Europe. And then Jordanis will explain all the background related to the Accelerator Challenge on novel biomarker-based assays. We will have a short break to stretch our legs, and then the question and answer will start, which will be moderated by Vilma. And of course, please, as I just said, put all your questions in Slido. Around 11:15, there will be 5 minutes closing remarks of Jordanis, and we expect to end around 11:20. So, my name is Annemarie Sasse. I'm the head of unit of the Program Manager's Office in the European Innovation Council. And we are organizing this week and also next week information days around all the challenges that we have in the European Innovation Council. So what is the place of the EIC in Horizon Europe? You can see it here on this slide. So the whole Horizon Europe is a 95 billion euro research and innovation program. It has the structure that you see on the slide. There are 3 pillars. That's the bulk of the research of Horizon Europe. The first one, pillar 1, is excellent science, and there you see the European Research Council, for instance. Pillar 2, it's related to collaborative research on global challenges and European industrial competitiveness. Pillar 2 in itself is again subdivided into clusters, and there is a cluster, health, which is of course very related also to the topic of today. And there's a lot of health research in pillar 2. But the European Innovation Council is placed in pillar 3, called Innovative Europe. And this word innovative is very important. The goal of the European Innovation Council is to bring the excellent research that Europe has to the market as soon as possible. And around 10% of the total budget of Horizon Europe is dedicated to the European Innovation Council, so around 10 billion euro. In the European Innovation Council, we range the full spectrum of research from early stage to bringing to the market. And this is represented by technology readiness levels. We have 3 instruments. The first one is Pathfinder, and Pathfinder is dedicated to early stage technology research. So from technology readiness levels, TRL 1 or 2, bringing it up to TRL level 4. This should be potentially groundbreaking research, but still very risky. And the instrument used is that beneficiaries will get a grant up to 4 million euro. So with Pathfinder, you validate early ideas. It could work. In some projects, already a business model is clear market perspective, and they might go up to higher TRL levels, and they can go immediately from Pathfinder to Accelerator. But there are also projects who, after the grant ends of Pathfinder, they need to mature their technology further, and they also need to develop further their market outlook. In those cases, we have the instrument called Transition, to transit from Pathfinder to Accelerator. And there you want to move up your technology from TRL level 4 to TRL level 6. So it's really to mature further the still promising ideas coming out of an earlier research grant. So there you also receive a grant, and that can be up to 2.5 million euro. And then the third instrument that we have, Accelerator, that is really for commercialization and scaling up the innovations. It is for SMEs, for individual SMEs who want to start this company. And the type of financial support that they get, it's blended finance. So there can be a grant part up to 2.5 million euro, but there can also be equity investments up to 15 million euro. So the financial ticket of this instrument is much larger than the others, because that is really the type of financial support that is needed to scale up the business of companies. On this slide, we see again the three instruments, Pathfinder, Transition, and Accelerator. We see, as I just explained, that the technology readiness level goes up with every of these three instruments as you follow them. But what we also see is that also the business readiness level goes up at the same rate. So in Pathfinder, the business is not so clear yet. It's still much focus on developing the technology. In Transition, there is the chance to develop this business outlook more. And in Accelerator, the business outlook should be clear. And the EIC, in that way, accompanies the beneficiaries in their entrepreneurial journey, going from research to business, where in research, the question that is asked is more what can this technology do for me? Can it solve my problems? So it's really technology development and validation. Whereas on the business side, you would be answering questions like, why would customers buy my products? How can I address their customer needs? So the question is different. It will be more a business question that you need to ask yourself, and you can use the financial support that we get to really get a good grip on answering this question. We do not only provide financial support in the European Innovation Council, but we also provide services to all the beneficiaries. And that is summarized in this slide. So we have the business acceleration services, which are available for all the beneficiaries. And you can see on the left-hand side of the slide all the services that are available. For instance, access to entrepreneurs. So the business acceleration services, they provide opportunities for companies to be in contact with investors, and they organize events where relevant investors can hear the innovations that the EIC beneficiaries have developed, and so through pitching, for instance. There are mentors for you available and coaches. There is access to ecosystems, for instance, in the form of corporate days. So we organize events where a corporate who would like to meet the relevant EIC beneficiaries in their domain And we organize there a matchmaking event with pitches where the corporate explains their needs to the companies can pitch what they have, in the hope that this then will lead to further collaboration after those events. So with the EIC, you can also extend your ecosystem. So there are many of those services, but we also have something very special which we don't have in the rest of Horizon Europe, and that are program managers. And today you will meet Jordanos, the program manager for health and biotech. But there are 10 program managers in the whole European Innovation Council. And on the right-hand side of the slide, you can see their expertise. They are hired for 4 years by the European Commission, and they are in-house experts. On the one hand, so they have a proven track record, both in research, but also in bringing research to the market. So they know the whole spectrum from early research to really creating innovative companies. And they do 2 things. First of all, they develop a vision on where the European Innovation Council should invest more. And they do that based on intelligence that they have. They know, for instance, where research, where there's a strong base of competence in Europe, but where we don't have enough companies yet. And they then develop a vision on how the European Innovation Council could invest in that area. And that translates itself to the challenges that we have in the work program. And then the second part of their task is, once the projects are selected that respond to these challenges. So if you submit a proposal and you have been evaluated positively and you get a grant or other support of us, then you become part of a portfolio. And then the program manager works with all of you and the other projects in the portfolio to address common issues which might hamper or which might make it very difficult for you to go to the market. But by working together with the projects and the program manager, the idea is that the impact of all your efforts will be much higher than if you would do this all on yourself alone. And that's what we call the proactive portfolio management. So this slide gives an overview of the whole work program of the European Innovation Council. This is the 2023 work program. In total, there's 1.6 billion euro of funding. And you can see on the left-hand side the 3 instruments. So we have Pathfinder with 343 million euro, Transition 128 million, and then Accelerator 1,138 million. And each of these 3 instruments, they always have 2 parts. About half of that budget is related to an open part where you can submit proposals on any topic on which you want to build your business or do your research. And there is a top-down part, which is then the part of the challenges, which is designed by the program managers. So each of the 3 instruments has this open and challenge part. And then if we go to the right-hand side of the slide, you can see all the challenges that we have in Pathfinder, in Transition, and also in Accelerator. And today we will talk about the biomarker-guided cancer treatment Accelerator Challenge, but we have been organizing information days about all of these challenges. And they're all made available as recordings if you would be interested in any of the other ones. So I want then to finish with an overview of the deadlines that you need to respect. So we are here now in Accelerator Challenge. So you can see that there are 3 deadlines that are relevant for you: 22nd of March, 7th of June, and the 4th of October. So if you want to respond to this challenge, you can do that at any time from now on. And we will collect all the proposals up to a certain cutoff date and do an evaluation of those proposals. So I also want to refer you to the official work program of 2023. That's the legal basis. That's what we call our Bible. That's the text to which we stick during evaluation. So this you should read very carefully. And I want to refer you to a recording of the general info day about the European Innovation Council, which took place on the 13th of December. And there, everything will be explained, for instance, on how to submit a proposal. Today, we will focus really more on the content side of this challenge, and we will say less about general issues about how to submit a proposal in the European Innovation Council and how your proposal will be evaluated, for instance. So if you need that information, then I want to refer you to this recording. So with this, I want to hand over to Danos, and so that he can go into the real topic of today.
Thank you very much, Anne-Marie. Welcome to everybody, a warm welcome to today's Info Day about the specific cancer call. While sharing my screen, I would like to also share with you information that in the area of health and biotech, that it's my responsibility. We don't only have action on cancer, but we also have action on cardiac diseases with other calls. We have action on synthetic biology, and of course on cell and gene therapy. So this is one of the 4 or the 5 pillars that we're working hard. But today, as you already heard, first of all, my name is Jordanis Arzimanoglu. I'm responsible for health and biotechnology at the EIC, and my background is I'm a medical molecular geneticist, having run careers also in management of science and as researcher at Cornell, New York, and later running bioclusters in Europe. This career brought me here at the EIC, and I hope to convince you after this presentation of the way we approach, of the intelligence we use, and of the potential application of all that we are striving here at the EIC. A new approach and with a lot of intelligence. So today's topic is about the novel biomarker guided treatment, cancer treatment. There's going to be a lot of science and science of potential clinical utility presented, and all that is to give you the background that you need to understand how we approach the topics that we are funding. I'll walk you through, of course, the scope of this challenge and the. Let me just try to take something from the my screen, and then, yeah. And then the specific objectives of the challenge, accelerator challenge, is at the accelerator level. The impact of what we are proposing at the clinical level, the impact at the industrial level, of course, the societal impact, and the very briefly a few words about the valuation. And is together with us my colleague, very much delighted to explain more if needed around the valuation. That's not so much the scope of today's presentation, as Anne-Marie said, but the scientific and clinical background is the number one priority. Some challenges that liquid biomarkers we're focusing are facing currently on their way into the clinic. What we have here in Europe at the EMA level in terms of regulation, these are all going to be very, very brief references for you. And of course, the main presentation would be around examples, around 10 examples of biomarkers under development with clinical potential. There is the importance, there is a focus, there is what we call the underpinning evidence on which, the grounds on which we have based this challenge call. And it, of course, is highly relevant with the health policies in cancer, as I will say at the end of my presentation. What is the overarching scope of today's presentation is, of course, to make a more guided, more personalized cancer treatment. And in order to do that, you need to have effective biomarkers. There is a lot of discussion and a lot of activity around biomarkers, different types of biomarkers. In reality, not all of them, a small proportion of them is quite informative. And this is the reason that there is resistance by the physicians to use them, exactly because they're not really sure about what they mean. And we need to increase all these biomarkers, informative biomarkers, where a physician can make an informed decision about a treatment, or how to avoid side effects, or what are the chances for recurrence, as you will be hearing with the examples that will be provided to you. So again, the overall is it what can we do to improve the integration of effective biomarkers into the clinic? And if I had to make it, more specific, we have 4 or 5 objectives. I would say the fifth one is not shown here, but has to do with if we're able to predict who among the precancerous lesions will be developed into cancer. Now, the first one is, of course, what everybody wants to know: identify who is more likely to benefit from a treatment. So far, in most cases, this is not known. So it's something that we'll understand later on after the treatment. But we would like very much to have this informed biomarkers to tell us who is more likely. My former clinical mentor at Cornell, he 30 years ago, 25 years ago, he used to work intensively and go into the clinic and always asking that to the young physicians. What do you think? Who is more likely? Who is more likely to benefit? Then is, of course, who is more likely to recur after treatment. So we are inviting proposals. We are inviting novel concepts. We are inviting people who are working with novel biomarkers that can answer those questions, can contribute to answer those questions. Then is the quality of life. So we'd like to have biomarkers that can help us understand uh, how can we avoid the side effects. That's very important. And we want to have, of course, biomarkers that can monitor the clinical progression of the disease. All these are 4 distinct categories of biomarkers that there are some right now that are used, but we need to do a lot of work to improve the situation in order to, in the era of personalized medicine, in the era of precision medicine first, eventually leading to personalized medicine. And of course, the expected impact is going to be at different levels. First of all, the clinical environment, and I will refer to that in a minute more specifically. Second is the bioindustry. There is a lot of bioindustry, small and big, that is working in this area. I would just like to inform that recent news from the National Cancer Institute in the States, 14 companies, very big size companies, are participating in a clinical study of 1,000 healthy people and 1,000 people with cancer, diagnosed and treated with cancer. And there is after many years of research, decades, there is a concerted action, I would say, to be able to identify the biomarkers. This clinical trial has just started. Leading physicians are running the trial from across the states, meaning timely our call with this clinical trial. There are others as well, but this is a very new one under the National Cancer Institute, are in line and are happening at, I would say, at the same time. And then is the also of the people that they want to invest in that, and they have a real interest in investing in this area. And the more activity exists, the better it is for investments. And EIC is investing in high risk, high gain, as you know, and is very much interested in supporting novel approaches. Now, with respect to the clinical impact that is expected, this is absolutely in line with the objectives. So if we have successful proposals, if we found successful works at the accelerator level, it makes sense to believe that we contribute to learn who among the cancer patients is more likely to benefit from a given treatment, what we call as guided treatment. Of course, when you have situations with all these precancerous lesions in different types, nobody knows who is going to develop and who's going to really progress more rapidly than the others. That is an expected impact. I'm always talking about the clinical level now. I'm at the clinical level. And then there is always a big question. We'd like to know if we have if we fund projects that are contributing to biomarkers that will tell us who is going to recur or not, that would be, as you can understand, a small breakthrough in the clinical practice. And of course, it's not only about efficacy, but it's also about the quality of life. Even for the people that are in the later stages in their life, we need to improve their quality of life. Meaning, there is no reason, if the biomarker says that there going to be no effect of a specific treatment, to be administered this treatment. And so far, the practice, unfortunately, has been that the protocols, all the people who were taken into heavy chemo or other treatments, and in the end, it was identified that many of those, they really it don't make any difference. So you understand that in terms of quality of life for the patient is extremely important. And that also couples very well with being able to monitor the clinical progression of the disease. Now, what is the difference here? This is a challenge at the accelerator level. There is a lot of activity, a lot of funding of within consortia of other startups from the health cluster calls. But what is missing here, and what we believe we can contribute, is that we want to create a biotech portfolio, a competitive biotech portfolio of companies working on biomarkers. So we're talking about not only startups, but scaling up companies, focusing specifically on novel biomarker-guided treatment. I would like to stress and emphasize the importance of a focused approach, of a targeted approach. In this call, in this case, is the treatment and not the biomarker-guided treatment. So if with this call, we want to, the outcome of this call is expected to be individual SMEs, companies that have this focus and share that activity to a greater or lesser extent. And this can become the basis of a future critical mass in Europe. I'd like to take 30 seconds to explain to you why we think this is important and why we think this is unlike any other effort that is happening right now in terms of investing. Because for the future European technological autonomy that we all envisaged, and you hear the politicians talking about that, you can't go for everything. You have to be very highly selective. So you need to select areas that you believe Europe has the knowledge, has the human skills, the people that can really drive and lead the way in those research lines and innovation lines, if I may say. And only by building those niche areas and strengthening these areas, and there's the role of EIC, you can create the critical mass that you need to be able to compete and sustain. I'm repeating, and sustain. This is very important globally. So this is the reason we go targeted. This is the reason we are very selective. This is the reason we are using intelligence behind and a lot of information proprietary when building these portfolios, in this case at the accelerator level of SMEs, to be able to compete globally. My colleague Vilma later on will answer if you have more specific questions. Basically, the evaluation process at the accelerator level is 4 stages. And the first one, I believe, is quite bold step that everybody at any time can send his ideas in 5 pages. I'm repeating, everybody at any time is very welcome to do that. So in a way, you are testing yourself, and many people say I have a great idea. So the question is, the EIC answer is you are welcome. Put it in 5 pages, make it comprehensive, and convince us that this is really novelty, and this is really timely, and this is really needed. Of course, if you go through this first step where the chances are not extremely difficult, then you have to build a proposal. We provide coaching for this proposal to help you, and this proposal, full developed proposal, will go to a remote evaluation, the third step, and then you will, if you go through that, you will be pitched in front of a jury, and the jury are people with knowledge in all areas that are covering the catalysis of breakthrough research to disruptive innovation to application. Key word for us is application, and key word for us in this case is the potential clinical utility. That's why it is at Accelerator. That is why we believe we make the difference with all the rest of the commission institutions when they are funding only research. Now, there is a focus in our call on what is called the liquid biopsy, the liquid biomarkers. Those are all the different tests that are maybe working when you take a blood sample and then you try to identify the biomarkers informative for a specific tumor type, for the treatment, or for the potential recurrence. But the problem is that with these biomarkers, there are several challenges that you can see here. Of course, the harmonization, standardization is a key challenge, and you need a lot of sophisticated pre-analytical analytical testing in order to be able to bring that to the clinic. There are no quality controls, and the evidence is sometimes is good, is relative. In very few cases, there is strong evidence what you need, which is absolutely a prerequisite to bring some to the clinic. The results vary, and as a result of that, the clinicians are very reluctant, to learn more or to accept that in the common practice. So there's a lot of work to be done, which makes this call very timely, and I would say I would tend to say also very much needed. We need to work, and then that is why we are putting this forward for applicants in Europe to apply. And what happens at the regulatory level? Of course, I'm talking of what currently EMA is doing, and EMA, of course, requires qualification of biomarkers, and this is very good. You have to go through qualification because otherwise you can't prove that your biomarker is good more than at the research level. You need to have it all the qualification tests. So EMA gives an opinion, meaning the applicants can contact EMA in the broader spectrum of scientific advice, but there is a specific office that deals with the biomarkers, and for all the applicants, for all the people working in the field, you can contact them to get, really very important information that is needed. And then the goal is to certify that your biomarker for a specific clinical use, pharmaceutical research and development is capable of going through the test that EMA will require. We learn more around that because we've recently organized an European Innovation Council, European Medicine Agency Info Day, and through that we had people from EMA describing all the spectrum of advice and activity that EMA is doing, and EMA is improving rapidly. So that's why I have put this EIC EMA Info Day. That was a few days ago. And we are convinced that we have to work closer with EMA. And even more important is not if we are working closer, is if the applicants, if the people that are working in the field of biomarkers, that they need to understand that they have to interact with EMA, because EMA is also needs the information from them of what is happening, what are the new research lines where regulation has to be developed. And the only way to do that is by informing EMA, and you can do that through us, the EIC as well. So this is very important in all these areas like the qualification of biomarkers, because the challenges are many, and they were listed in my previous slide. From now on, the rest of the talk will be the examples in the background, the scientific background that is needed and is important for you. We believe to understand why we're putting this challenge forward. The examples are for predictive biomarkers, prognostic biomarkers. When we say prognostic, we mean favorable or non-favorable. Companion diagnostics, which is, of course, given together with the treatment. And all this information that you will hear from different tumor types are in support of this challenge goal. The first one comes from bladder cancer. Allow me to, yeah, okay. This is the case of the circulating tumor DNA is something that, of course, you can find in the blood as a biomarker. So it is known, these are all works that are presenting in top journals, and this is all research with clinical potential clinical application of the last 2 years. So it has been associated that if you find even traces of circulating tumor DNA in the blood, so it is more likely that these patients will relapse. So these patients will benefit from immunotherapy. There is no need with works like that are paving the way for a biomarker to give immunotherapy to everybody, but only those that will be tested, and circulating tumor DNA will be identified in their blood. So with my first example, all this underpinning evidence I'll be presenting to you, I want to bring you into this thinking that says that you need to stratify, you need to group patients, you need to understand all the different indications, or hopefully all the documented evidence, scientific evidence that can allow for the stratification of patients and knowing who is more likely to relapse and who is not. So this is the case here with the bladder cancer. Let me go to the next. The next example is perhaps many of you know this company. It was founded by us 10 years ago, a very successful Dutch company that is in many hospitals. You will find their recurrence tests, the MammaPrint. There are actually 2 MammaPrint Oncotype. They're used clinically. So they did what is called a meta-analysis, and this test has been in use for many years. And they studied 7,000 patients. That was a study of over 10 years following these patients. What was striking, and they realized that one of two patients should never have received chemotherapy. So this is also an example to tell you that the quality of life. That was realized after following these patients, 7,000 patients is a very big number for over 10 years. And we know now, based on this analysis, and that is to create this kind of thinking, that how important it is to analyze and knowing and develop biomarkers, that one of two, like over 3,000 patients, there was no reason at all to have received chemotherapy, however they did. Now, if we go to another tumor in the Barrett's esophagus, another clinical pathological situation, indication, then analyzing, comparing 40 patients and 40 with stable Barrett's esophagus and 40 patients who progressed to adenocarcinoma. In the beginning, the mutational analysis would lead to nowhere. There was no clue. So there was no clue why some patients progressed to adenocarcinoma and others they didn't. But when they deepen their research, you see always the slides, the publications from Nature or other journals on the other side. When they deepen, they managed to find that there was one specific cell subpopulation with a specific expression of structural variants and other high-level applications, which could be used as a biomarker, and I'm sure they're working on that, and to really inform who is more likely to progress to adenocarcinoma. Another more important thing about that is this specific characteristic biomarker was detectable even 6 years prior to cancer diagnosis. This challenge focuses on treatment. Guided treatment is not focusing on cancer diagnosis, but you all can understand that information, critical information, contributing to the development biomarkers can be used even beyond treatment. And this is one example that the specific potential biomarker is detectable 6 years prior diagnosis. And this was the only difference in the end between the two groups that are compared of Barrett's patients with a specific clinical pathological phenotype, why some they go and develop adenocarcinoma and others not. Another example is, and many people talk about that, and my former connections with former colleagues from long time ago at Memorial, they're advancing a lot on that, is everybody would like to have the possibility to work also from the regular standard pathologist light. And of course, artificial intelligence can help a lot, but sometimes people tend to exaggerate, especially those that they come from not the medical field. Artificial intelligence can help if you know the pathway, if you know what are the actionable mutations, if you really know the biomarkers in the that can be informative and not relatively informative, or you have strong evidence, which is, again, I'm repeating the goal of this challenge to contribute to that. So it is important to be able to act clinically with information that comes from the pathology slide, and biomarkers there would be extremely helpful. instead of having, to the patient to undergo post-biopsy treatments and all kinds of tests. If you could act with the information from the slide, that would be great. And of course, all this research in this area needs to be validated and deployed at scale before it gets to the clinic. Another example from a different type, breast cancer in this case. And of course, there is a lot of work has been done and trying to subgrouping and creating subtypes in breast cancer. This is a work that I want to present to you because the emphasis is on the different types of multi-omics analysis. And it's, of course, a huge consortium. The goal is to try to associate, not correlate. Correlate is not enough. We start sometimes with correlations that are observed between molecular and clinical phenotype, but in the end, you need to establish what is called, from a genetics, a real association. So they're working on 10 different predictive biomarkers with 10 different cancer treatments. You want to match the biomarker with a specific treatment. That is the goal in order to have an informed clinical biomarker and to increase the chances to bring it to the clinic. So I don't want to get into details, but I'll tell you that all multi-omics are used, different types are used, and there is a lot of work in this area. As I said, it's not only if something is efficacious, it's also the quality of life. So can you predict the side effects? It seems that with the biomarker that this group is developing, which is at the RNA, at the transcription level, at the gene expression level, and looking only at immune cells, in this case, T cell subpopulation, not just cells, but T cells and the subpopulation of cells. I hope that I'm giving you information to understand how deep now it goes and how specific it becomes. Then the specific gene expression particles have been identified at these T cell subgroups that can predict who is likely to have adverse effects. And this has been investigated 4 to 6 weeks after treatment. So if you remember, in one of our among 5 objectives was to be able to contribute to this very much unmet medical need, to be able to know who is likely to really suffer from the treatment. And this work here, presented here, is in line with that. A specific T cell subpopulation can tell with quite a good specificity that the patient is likely to suffer severe side effects from treatments that are shown here, like the anti-PD-1, which are now in the clinic. We talked about at the gene expression level. Now we're talking about, in this slide, about inflammatory biomarkers. When I'm talking about inflammatory biomarkers, is a type that biomarkers that, of course, are increased when you have inflammation. The example here is with interleukin-6, with soluble tumor necrosis factor, and the CRP, which is very well known. So what is the important from this study, and why I'm sharing that with you? Because you can distinguish, you can subgroup patients based on, if you have this in higher levels of these biomarkers, there was association with increased risk for recurrence. Remember again, the third or the fourth objective of this challenge is we are seeking for novel biomarkers that can help us be able to make clinical decisions who is likely to recur or not. This work is exactly in this line. And this work is not at the DNA level, is not at the RNA level. We're talking about specific molecules that are identified in inflammation. So you can use those biomarkers now at this level, the inflammatory biomarkers, to be able to associate the association method. You can use that to say who is more likely to recur or not. So we think this is an important work that shows the wide range of biomarkers that we want them to answer the known questions: guide the treatment, recurrence, side effects, and clinical progression. Another work here, now we're moving to proteomic level, immune proteomic level. As I said, again and again, there are different types of biomarkers. So although our call is very targeted to help and guide treatment, the range of biomarkers can be used is quite significant. So we're expecting and we're hoping to receive applications that will cover different areas, not only different tumor types, but different types of biomarkers. So if you couple those things, the activity that exists is very, very significant. So this example here is from gastric cancer. And the important thing that I would like to draw your attention to, we're talking about the biomarker that can inform before surgery, ahead of surgery. This is another clinical challenge here. So through this work, it was identified that you can, by testing for proteomic profiles of immune cells, you can tell before surgery. It's these people who are receiving chemotherapy and those before surgery, who is likely to benefit or not. And that raised the issue that perhaps we need to change our view and go for a more systemic immunity testing prior to surgery. That example here is from gastric cancer. And it would be clinically beneficial, a patient stratification strategy at the level of proteomic markers. I'm repeating again, proteomic biomarkers, in order to know the prognosis of those patients before surgery. So we're talking about prognosis before surgery. Other examples that I showed you were after surgery, and you wanted to know about, again, the recurrence and the relapse. A variety, quite a choice, I would say, where you can intervene with a guided treatment before or after treatment and make informed decisions. And for instance, I'm coming back to bladder cancer to say that there is a lot of work because the research so far, the clinical research has not really produced the expected results, which makes us believe that there is a lot of room here for novelty, a lot of room for new works. The example here was to show you that although in the US the testing is recommended and is happening, in the case of bladder cancer, only 5%, 100% undergo the different types of testing, but only 5% can benefit and only 5% can receive targeted therapy. So the work so far has not been very productive, and there is only one targeted therapy approved in the States. 95% of the patients undergo screening, but you cannot do anything. This is not helpful because there is no evidence that you can match what we call actionable mutations, specific molecular profiling that to have evidence that a specific clinical phenotype will be developed with targeted therapy. So this 5% is something that makes us believe that we are not only timely, this is very much needed. We need to do something for the rest of the 95%. And what the information I'm bringing to your attention here is that there is only one targeted therapy approved for this for bladder cancer. If we go now to lung cancer, which is among the biggest problems that we are facing right now. Again, we want to make a lot of, there's a lot of room for improvement. We haven't been successful at all in a way to know what are the actionable mutations. Again, remember, actionable mutations, in this case is lung cancer, and this work is with trying to identify the differences between adenocarcinomas and squamous cell carcinomas in lung cancer. Some of these biomarkers are known for years, yet not good enough. We need to do more. We need to go for novel biomarkers, or we need to find, the combinations that can be interpreted and brought to the clinic. For instance, for one biomarker, which is the third point in this slide, there are the variations in the expression or at the DNA level of this biomarker that are seen between but the clinician can be informed, but is not really, how can I say, implemented the clinic for the reasons that I mentioned before. A lot of work, a lot of effort, but we're not there yet. We need to the point to have these actionable mutations for sure and to implement them in the clinic. So this example comes now from the lung cancer, which, by the way, is identified as the #1 in Europe in a recent meeting that happened in Sweden, and my colleague Jenny Todorova informed me this morning, where the Commissioner of Health was there and the ministers so from Sweden and Spain. And the goal of the meeting, when it was identified that lung cancer is really top, it was to compare the cancer profiles between different member states. I'm taking this opportunity to mention that to you. I believe it's on the news today. And what was identified is that we have a twofold difference between member states in Europe. So we need to do a lot of molecular work, a lot of proteomic work, a lot of inflammatory work, but we need to do some harmonization between the member states. It's really striking to have twofold difference between member states when only in 1 year 200 billion are spent for healthcare in Europe. Now, let's go to another type of potential biomarkers, and in this case, the example is the AML, acute myeloid leukemia. It's interesting. We're talking about a three-dimensional biomarker. This is relatively more new, I would say. This work comes from it's from last month or the previous month. So a new field here, a new type of biomarkers. When I'm talking about 3D at this level is changes in the chromatin structure, of course, coupled with methylation patterns. And with these biomarkers, you can, there is quite a hope that this can guide treatment. So changes in at the 3D level have been associated with with this change, they have been able to subgroup AML patients, and there is quite a good hope that, of course, you can match specific treatment based on this subgrouping of AML patients, depending on this 3D genome analysis testing biomarker, I would say. And that is, so much in line with the purpose of this call, to be able to guide and influence the treatment of AML and not to give the same treatment to everybody. I think that's the last example. And when we're talking about novel approaches, and when we're always saying to you that EIC is about high risk, high gain, and about, I would allow me to say, the most crazy things or totally new approaches. One example I would say is here, where microbes, yes, microbes can be used as biomarkers. There is a lot of work is underway, ongoing, that is relating different types of the GI cancers with specific patterns of microbes. And this is a promising area, an area that somebody would discuss very generally or wouldn't think that would be a favorable area to work. But when you have evidence like the one that's provided here, for instance, that for stomach cancer, not in general the microbe pattern, but one specific fungus, the Candida tropicalis, when is identified, the evidence says that these people are very, very likely to die and have the worst prognosis. It opens the door of other people to want to investigate and try to associate specific microbes and specific profiles of the microbes with one or another tumor type. And you see there's quite a variety. The work now is going from all the GI, but it goes also to lungs and breasts and all the GI tract. So this is a relatively new field, and we are excited. I mean, that if people are working or people want to work on that. I think is enough we have of the examples that is a background of what we are putting forward here in this challenge. But it was our obligation, it was our duty to look into what has been funded so far. And the colleagues from the Cancer Mission, from the DGRNI at EIC, they helped us in doing this systematic analysis. And sometimes just by the title, you think tremendous overlap, but this is not the case. We are very focused and we have looked into that, and we are not overlapping with other, with either previous works, previous funding, or current funding. And we looked, of course, also at the goals that are supporting by the Innovation Health Initiative. And in one case that we went into deep, and again, we are very happy that, for instance, there is emphasis on the imaging. We are focusing not on the imaging, but on the liquid biopsy and other biomarkers. So what I want to share with you is that we do scrutiny, when we go for an area to make sure that this is novel, this has not been funded, on top of the fact that this is very much needed, hoping that I convince you about that for all the scientific background that I presented. We are very pleased that one thing is not to overlap, another thing is to serve other purposes. Cancer mission now is one of the 5 missions in priority for Europe. And together with my colleague, Jan van der Loo, who contributed a lot to the preparation of this call, we checked, and he actually, with his experience in the field, and he's member of the cancer mission at the JRNI, we both agreed that here we have a complementarity, and we're very happy because one of the objectives is absolutely in line, complementary to the goal of this challenge. So this challenge will also serve the purpose of the cancer mission, and I'm providing here the evidence specifically on objective 3 of them that is described in the implementation plan of the cancer mission for Europe. That is now a priority, European priority. My colleague Anne-Marie has presented all the different links here, so I think I'll stop here. And before we get to the questions and pass the floor to my colleague Virma Leite, I would like to invite the director of EIC, I believe is with us, to
Yes.
pass the floor, and we're excited to have you with you. Please share the views or with your experience. EIC, I'm sure all people know, international agency for research cancer based in Lyon, and Dr. Elizabeth Wirdepas is with us to share her views. We are focused on treatment. EIC is very, very well known across the world for the work in cancer prevention. And please share with us your views on what has been presented and the timely nature of this.
Thank you. Thank you, Jordanis, for the invitation to attend this information day on the European Innovation Council Acceleration Challenge on novel biomarkers to guide personalized treatments. As you said, I'm Dr. Elizabeth Wirdepas, the director of the International Agency for Research on Cancer of the World Health Organization, and we are based in Lyon, in France. So based on our estimates for year 2020, the global cancer burden is projected to rise from 19.3 million new cases per year in 2020 to 13.2 million in 2040, and from 10 million deaths to almost 17 million deaths in 2040. So a massive increase worldwide, and 20% of those are in Europe. So the so-called market is much broader than Europe, is 5 times broader. So this is so important for us to keep in mind the needs. So these figures are really alarming, and I think this calls for the science communities to work together. Although the greatest increase in incidence and mortality rates will be in low and middle income countries where cancer services are already hard pressed, even the richest countries in Europe and elsewhere will struggle to meet demands of increasing cancer patient numbers and spiraling treatment costs. Advances in molecular therapeutics in the past decade have opened up new strategies for identifying who is at greatest risk of developing cancer and for determining which treatments are most likely to benefit cancer patients. However, there are difficulties and unresolved issues in the development and validation of biomarker-based personalized therapies. Further development of these strategies is in an efficient and timely manner requires investment in the discovery and validation of molecular biomarkers with predictive risk and prognostic values to improve early detection of cancer and guide personalized cancer treatment. At the International Agency for Research on Cancer, where I work, research on early detection biomarkers to optimize personalized screening is very dynamic, with many promising new candidates such as blood or urine-based biomarkers, liquid biopsy biomarkers, circulating tumor DNA, as well as imaging biomarkers. This will result in a selected panel of biomarkers of cancer risk for prostate, lung, esophageal, bladder, oropharyngeal cancers that will be incorporating risk prediction models and used to identify those subjects most likely to benefit from screening and early treatment. To make effective progress in this area, we would need highly targeted calls on the validation of these early detection biomarkers and the demonstration of their practical clinical implementation and their utility for clinical decision maker. Today, this call target on biomarker-based assays for personalized cancer treatment is really very relevant and very timely. Relevant because highly targeted calls in cancer is of the utmost importance to reflect on concrete unmet needs in this area that need to be tackled, whereas broad calls fail to identify clear research or biomedical focus. The design and development of a potential targeted and impactful call is likely to make the difference. I used to say that research that matters is what we need, that will benefit directly people and cancer patients. So back to you, Johannes, and thank you for this opportunity to address the audience.
It was a pleasure, Elizabeth, and great comments, great input. I loved the research that matters. That's it. And I just like to. It's so important that for all of us and for the people to listen that the activity that is under ongoing at EARC. EARC is an international center with proven track record, and it's very reassuring for us to have your views that this is really timely, and people that working right now in React and they want to get into the innovation journey can try also their luck through this call. But even the broader picture of that we have these activities in Europe, we have the pleasure and we are fortunate to have EARC based in Europe, which give us a lot of possibilities. Thanks again for your intervention here, and it's a pleasure to have you with us. Now, I think it's time to take a break before we get into the Q&A session. So we're ready to start the Q&A session. I'll pass the floor over to colleague Wilmar Adelaide for the questions.
Yes, hello, thank you, Danos. So indeed, we have a list of questions from participants, and we already answered to one in writing, and we will go through now one by one. They are technical questions, specifically addressing this topic. So I think this is a very good, let's say, feedback from audience to clarify in more detail, and I hope it's probably together we will be able to help potential applicants to understand better. So could we maybe start with the first one?
The first one about the digital biomarkers, right? If I understand.
Yes.
Yes, the answer is yes. I brought the example with the AI with a slide. So yes, the answer is yes. The second one.
Well, the question is about preclinical validation of the challenge. You comment on that?
What I want to say is that when you have a proposal that talks about a clinical phase 1, they need support for that. But at the same time, in this proposal, somebody wants to validate further with a higher number of samples, which is the case, and this is what's happening in reality. You cannot shut the door and say this further validation step, is out of context, and if that is in the context and the main proposal is about the clinical trials to prove that, then this is the reason. It's not that we include the preclinical validation. We're talking about an EIC Accelerator Challenge. Is that sometimes the work that is in front of us that is suggested it makes sense to have this further second validation at a high number of samples or a high number. So you can't say that this is something that be excluded, as long, again, as long as the main focus of the proposal is on the providing evidence of the clinical stage.
Thank you,
No, the next one is, of course, no. All individual submissions to address all 5 specific objectives. No, of course not. And this is, I would say, this is impossible. In a way, it's one group works in the recurrence, another works in finding the best treatment. The other one may be focusing on reducing the side effects. So it's very unlikely to work on all 5 objectives.
Next question is about the jury members selection, particularly for the challenge. I could probably address that partly because currently we are reviewing the jury members for this year, upcoming evaluations at all the stages, including also for challenge-based scores. So yes, we will have more, let's say, targeted list of juries. And just to add that, of course, program managers will be also involved in this review and to help us to select the best ones for this very important exercise.
The drug development is not the focus of this challenge. If I'm reading correctly the first question, the drug development is not. We're talking about treatment here and informative biomarkers. Now, if because of a subgrouping or because of the work towards developing a biomarker, a new potential target is identified, that is no problem. But if the proposal focus on drug development is not in scope with this call.
Here we have a question about preclinical stage. Danos, would you like to add something to what you said before? Or?
I wanted to say for all the questions regarding TRL that, this, allow me to say this difficulty, this sometimes confusion is from the fact that TRLs is something that comes from the engineering field, this readiness level, and this cannot be fully translated in health. And we need to improve that in the future. We have a very good definition of the different TRLs level, but I would always say the importance is on what is the concept, what is the novelty, and not so much, being stressed or being, how can I say, concerned with a TRL. In the health world, we're talking about preclinical, clinical, validation. These are the terms that we understand and the terms that we are using. And of course, this can be, they need to be aligned with the TRLs. So first of all, is the definition of TRL. As I said, we have in-house definition of the different TRLs, support preclinical validation, or I think when you say you are at higher TRL, if that means that you are at clinical phase 2, is that, I'm not sure what is meant. If you are higher than clinical phase 1 and you're at phase 2 and you go for blended finance, for instance, which covers a number of patients that can be included in phase 2, yes, that would be possible. But if it's a disease, it's not an orphan disease, a very big disease, I don't think that the funding from us can cover phase 3, for instance, but phase 2, in certain cases, it would be appropriate. What is the next one, Vilma, please?
Also addressing TRL levels and which TRLs, so you were mentioning several examples in your presentation. I think this question came during your presentation, and the question is, the participant is trying to understand which of TRLs in your examples are you in our call, what are we looking for? And is it compatible with TRL 5.6 as required by the accelerator? So indeed, I could already partly answer to this question. This challenge-based goal is part of accelerator. So yes, this is compatible. And Danos, I leave it to you to go into more detail about the TRL level.
Yes. No, it's, as I said, is what is important is that you include, if there is further validation needed, and that validation would be at the what you call the preclinical level, together with a solid application of that you are seeking funding for clinical evidence. That is okay for us. The emphasis should be at the clinical level, and that it would be at the TRL 6, I would say. Again, oh, we missed one question. I'm sorry. Oh yes, the medical imaging and magnetic. The focus here is on yes, the liquid biopsies, but I presented different types of biomarkers at the proteomics level, inflammatory level. The focus, imaging, medical image is not excluded, but the focus is not on magnetic resonance and computed tomography. We would like to have AI in proposals that is helping to improve the different types of biomarkers with emphasis on the liquid that I presented today. The focus is not on magnetic resonance in this call. That is for sure computed tomography. But there is imaging, that goes beyond that. And if there is, bioimaging and can be used as a biomarker, this is not excluded. But again, the magnetic resonance computed tomography is not the focus of the call.
The next question is asking why this challenge was placed on the accelerator, but not the transition, because I don't know who asked this question. It seems to this participant that both TRL and business readiness level may fit better. What would you answer to this, Danos?
From the very beginning, I said the scope is the clinical stage. In the second slide says we need the clinical evidence and we want to provide the clinical. So based on that concerns the vast majority. This is why it is at the accelerator level. Again, we should not be so much concerned about dividing in the health sector. I'm talking specifically in the health sector here, but we need to really shift the importance. So do you have really something novel? Do you have really something with clinical utility? Do you have the evidence that is required? This is important for us. You need to have enough preclinical evidence in order to substantiate and in order to document that you want funding for clinical work to bring to the clinical stage. If a little bit of that in the proposal is some further validation, that won't harm.
I would also maybe like to add that in Accelerator, there is more opportunities financially because you can combine it with equity component and we can follow you throughout the longer stage of your project. So that I see also as an advantage. Well, the next question is again technical. It's for you, Danos, about the guidance available on how multi-maker or omics approaches can fulfill the requirements of the in vitro diagnostic regulation. Could you comment on that?
Yes. I don't know exactly what the guys on this refer here. And as you know, and we are very fortunate to have this session with Emma lately, IVD right now is out of Emma scope yet, right? And the MDR is only if this is coupled with a biopharmaceutical. If you don't have these conditions, then you go out of the regulatory interest, the regulatory guidance that you can get from EMA. Is guidance available on how multi-marker approaches? If the guidance is referred to the regulatory path, or I'm sure it does to that, I think I answered that what is of EMA concern. The rest is notified bodies, and the rest is the CE mark and the other avenue. Right now, I'm just repeating, where we stand is that EMA is setting some pilot studies to see into more and more incorporation of what's happening in the medical device area because of all these delays and all this confusion that is out there. But this is at the pilot phase. When we have IVDs, is not EMA. When we have MDRs, is EMA if only this comes in a project that you want this device for a biopharmaceutical application. This is clear, and you can have all these answers if you follow the presentations that experts from EMA gave in our event, which happened on 3 days ago, January 31st. Clear, very clear presentation about scientific advice, about what they do with IVD. Yes, when is it for them to advise on MDRs? Only when they are incorporated with the development of a drug or biopharmaceutical.
The recording of this event online. So
I can't hear you.
Now there is another technical question about the challenge, about the scope of a challenge, and would this challenge cover the new diagnostic test based on already described biomarkers?
Yes, that's a very good question, actually. We are seeking for novelty, other novel biomarker, I think it was clear, or novel concepts, which means that if you use existing knowledge, of course, but then the further development has to have the novelty, the element of novelty that we seek in order to bring this, to develop it further, to investigate and to make it something of potential clinical utility. We are both interested in novel biomarkers and novel solutions with existing knowledge. There is no way one can discard the existing research. That's why I took the time and I presented to you that there are areas, there are tumor types and biomarkers of different types that already a lot of work has been done, but we are not there yet. So the next level is subject to this call, but the next level when it comes to us as a proposal has to have a clear novel approach, clearly something new that needs to be experimented right at the clinical level in order to tackle the challenge and make this fruitful and hopefully contribute to bring specific biomarkers that have been for many years around investigation, but not enough evidence to bring them to the clinic.
Thank you, Danos. Next question from Elena is also about the scope of the challenge. And she's asking, is a platform for drug development for personalized cancer treatment or for treatment prediction fitting is covered by this challenge or not? Do you comment?
It's the treatment prediction is the key here. We need to have, that's why I said, it has to be associated with treatment. And the goal here is not prevention, is not diagnosis, is the treatment. Any biomarkers prior to treatment, after treatment, that's why I described also the example of before surgery, that you have some informative biomarkers telling you that somebody is going to have side effects or in other cases, the surgery won't be as helpful. We need to focus of the association with treatment. So if there is a association with treatment, of course it fits. That's the answer.
The question about is the focus of this call, of this challenge, mostly on liquid biomarkers, or is cancer tissue-based biomarkers also of interest? And I think we had a similar question. Please, Danos.
Of course, it is also the cancer tissue-based biomarker also of interest. There are many examples. It's not on liquid, maybe one of the most intense areas for understandable reasons, and the director of EARC said that you're using not only blood but other fluids. It's non-invasive, so it makes a lot of sense. So the possibilities you have there, but also other cancer tissue based biomarkers. Of course, they are very much of it. They are very much welcome. I believe there is another question on top of that, or
yes, we still have a participant who already completed clinical evaluation, and they would like to work on clinical utility studies and go to the market tasks. Would you suggest that such applicant would apply under the challenge, or better to be open?
Would you like to take this question, Vilma?
Both challenge and both open call would provide you opportunity to apply for grant component if you still have remaining pre-TRL9 activities, and also for the equity component if you want already to, and you are close to the market. For the evaluation purpose, the scope of this call covers all the stages, right, Danos? I'm looking at you.
That is correct.
You
yeah.
are the godfather of this call.
That is correct. In order to answer, but you said it already. To answer, more specifically this question, it has to be said. We have completed clinical evaluation means what, and want to go the clinical utility study means what, and then we said that we can go and with a blended finance, we can go and cover also clinical phase 2 in certain cases, but not beyond that. It is understandable. The size is huge. In some cases, clinical phase 2. So if that is translated to what I'm saying right now, I think the answer is evident.
Questions are still coming up. So we have another one about scaffold technology mimicking tumor microenvironment for cancer treatment. Would that be covered by this challenge?
As long as the technology in the tumor microenvironment and I understand, I know the area, it can be the concept of what is proposed one can show the potential of informed decision that can be made based on using that within the broad context of the biomarker. If this technology can provide us some clues, some insights, even insights, because this might be entirely novel, that would, really support of having some clinical work in that area, I would say yes. But it has to be evident, not just the way it is. The way it is in the question now, I cannot see the potential, I don't see the utility, I don't see really. But if this is associated with the potential to be used for predictive or prognostic purposes, yes.
Thank you, Danos. And this was the last question. Indeed, we had a lot of very technical, and I hope we could cover them. If there is any last remaining question which our participants would like to ask, I invite you. Yes, we have a question about the recording, about the session. Yes, there will be on the website, which was dedicated to those info days for each challenge. You will be able to rewatch this recording. And we have another one. Question: if we decide to apply under the challenge, but the fit is not good, is our application moved to the EIC open pool? Good question. I would invite you to. No, I don't think there is a possibility for such a move. But I would invite you to read the work program for clarification, and we could probably answer you in writing. But you will come as anonymous, so it's difficult to do that. We have one more question coming in. Is it important to start phase 1 clinical trials at the beginning of the project? Danos, how would you suggest?
It is important to start phase 1 at the beginning of the project. I'm not sure what exactly is meant here. To start phase 1 at the beginning of the project. I mean, if, as I said before, if that means that if you apply for funding for phase 1, but also there's an additional validation before that, if if you have such a part, that wouldn't be a problem as long as the focus is really on at the clinical stage. Otherwise, I don't know how to interpret this question exactly. What it means at the beginning of the project. The clinical phase, the funding for the clinical phase, the clinical stage work is at the core, where at the accelerator level, this is obvious, we say it again and again. To start phase 1 at the beginning of the project, yes, you need to start the phase 1 with this funding. Huh?
Okay, I would like to go back to the previous question. I had a second to think, and just to answer, we are not moving projects from open to challenge or challenge open. What happens, because we have the same cutoff dates for challenges and open, and the interviews take at same interview week. But what is very important, when you apply to the challenge, you have to address a challenge. So the objectives of a challenge have to be present in your application. So no, it means that if you are not addressing the challenge, so you cannot fit under this specific
Call.
Yes, thank you, Danos.
So no more questions, Wilma, or?
There is a question about the link to this recording, but that will be available on the EIC website.
It will be available on the same website, on the same page where the info day it is. It will be available next week or the week after. Thank you.
Thank you.
If we don't have any other questions, Vilma, can you confirm that, or Robert or Jenny?
Did I confirm we don't, and so maybe we can wrap up. You could maybe
Okay.
discuss.
No, I'd like to close by saying only three things, and I'm very fortunate with EIC. We had the opportunity to share our views, the way we think through this call in this particular area. So the first thing is this call, as also other challenges from EIC, they would like to contribute to have transformative solutions in the area they are focusing. What does that mean? It means that we want to fund and we want to support, not only with funding, but also with intelligence and the services that come together at EIC, solutions, novel concepts, and novel biomarkers that can make the difference. So we take risks, and because the goal is to have not just something that could be of some relative potential use, but something that would make the difference. And this is, of course, risky, but this is difficult. But the overarching goal under the concept of the EIC challenges at every instrument is that proposals that can, that can come to us with convincing evidence of potential transformative solutions. Allow me to repeat the term transformative solutions. This is exactly, our main focus and what we'd like to see. The second comment is we do understand that people may be confused exactly on how much preclinical work is needed in order to try your luck at the clinical level. Nobody can answer that exactly. As the director of EARC said, there is so much work, for instance, in established and renowned institutions like EARC, a lot of biological fluids, not only blood, for liquid biomarker and others, and including tissues, and that because although there is intense work in this field for years, it hasn't paid off as we would like to have, as we would like to see in terms of clinical use, and that's the reluctance of the clinicians to really engage into that, or if you want to get the proper education, because that needs education, is new information when, and that has to go, of course, through the regulatory authorities. That's our collaboration with EMA, and that has to go through this what we call the convincing evidence. This is very important. Some people tend to work on existing patterns or existing biomarkers, but we have to sometimes think also a little bit outside of the box and try new approaches. That's why I brought examples like the microbe approach or other approaches to do the synthesis. And the importance here, another element that is that we are very focused, although different tumor types can be involved in these proposals and can apply, quite a wide array of tests at different levels, from genomic, gene expression, proteomics, that's also labs, inflammatory, all that is within the scope. The story that you are bringing to us is the important thing. Again, going back to the first point about transformative solutions. So this is why we are very focused, and this is why we take risks, and this is why we believe that we need to do that in order to increase the chances for European applicants to make the difference within, as an outcome of a challenge call. I'd like to stop here, closed up, thank all my colleagues for the organization, and above all of you that who attended. I'd like to thank Anne-Marie for the introduction. And of course, last but not least, a very big thanks to the director of EARC, Dr. Videras, for her contribution to to today's session. We hope to receive very good applications from you.